input to our project is the ligand. This website right here allows the user to customize and build their ligand. Let me just build a random molecular structure right here. As so, add some nitrogen, and then this ligand, we convert it into something called a SMILE, which is a code for a ligand. You submit this and you get this code, which is the SMILE. So a SMILE is basically this like universal like code for a ligand. Then we run it through this massive database of already existing SMILEs and their corresponding ligands. So since this is custom made, it doesn't exist, right? It's brand new, something the user inputs. Then we get this custom new SMILE. Then we go to existing database to find already existing SMILEs and their corresponding ligands. So what our code here does is that it's an algorithm that goes and parses through this entire database and finds the most similar SMILEs and the corresponding ligands. The point of this is so that we can find the corresponding proteins to these SMILEs and test that protein with the custom ligand and the custom SMILEs. So let me just run our code to show you. So the UI will open up, you enter the custom SMILE, which again we got from the SMILEs translator. You enter that here, you run it, and you get the 3 most similar SMILEs. So these SMILEs are already existing SMILEs from this database, and these are the 3 SMILEs. It gives you the similarity percentages, 34.4%, 32.6%, and so on and so forth. And then these already existing SMILEs we can use this to find the corresponding protein, and then we can use that corresponding protein and check how it reacts with the newly made SMILE and the ligand from this.
To try and find the type of predictions that we need to get from these oxidoreductase reactions, we need to first try and model the active site. So what we have here is a molecule from the PDB database, which is basically the protein data bank that is published by the NIH. This database has a collection of proteins that are known with their 3D structures and how they look like. So first what we want to do is we want to hide the cartoon, and as I've already made, I've made the active site and modeled it in PyMol. With this active site, we can see all of the binding residues with the polar coordinates. We can see all of the open atoms such as carbon, nitrogen, sulfur, which are all labeled in these different mismatching colors. This type of variation will allow us to see how the different molecules bind to these substrates. So it's a lot of processing, but if we can make a way to bind to each one of these open active sites. So you might be wondering, how does this PyMol structure link back to the similarity sequence that we built in the first part of this video? Well, this similarity sequence is only the first part of this equation. This similarity sequence takes us to a different database that we have made hand-built that shows us the 3D model active sites and how they bind to each of these residues. The similarity sequence will basically show us which atoms are different in each column with the SMILES format, because the SMILES format shows us with how the parentheses, equal signs, columns, semicolons are all arranged, which show us the general active site and how the molecule is fixated inside of its 3D molecular structure. So if we can model that, and we already have with our 3D modeling database, we can compare any molecular structure and how it'll fit inside this active site and give off any residues that it has. And basically residues are just amino acids that are labeled here, and these amino acids are the ones that are binding to this ligand. What happens next after we bind and find the active site and how it fits inside this kind of active site residues, right? So what we do next to try and model the type of reaction that goes on with every single one of these open exposed molecules and residues that are open to the 3D molecule ligands that the user builds is to use the tool IBM Reaction for Chemistry, which is basically a chemi-informatic library, which is basically just a library that has knowledge of chemistry reactions and we imported that into PyCharm, a coding tool. With this, we used the predict forward reaction tool to try and model how each of these reactions will react, and then we used a separate database that we found from the PDB to try and figure out how each of these molecules will come together with the reactions and how the final product will look like. So the final product will just be composed of each of these separate reactions that we then link together to create a sort of a com- composed product and molecule that the user interface can see from the originally inputted ligand and protein. So this is our entire project. I hope you really enjoyed it, and yeah, thank you.